Hi everybody, my next topic is the cutaneous senses. Here are the learning objectives. I'll talk about the somatosenses and the cutaneous senses. There are some differences, there is some overlap there. It is important to know that. Notice that these are all plurals. I'm not just talking about the sense of touch. We're way beyond that. There are two competing theories of sensory coding. Once again, it is specificity coding and pattern coding. We've seen this kind of over and over and over again, reappearing in other senses, and it comes up again. I wonder what the answer is going to be. Which one is correct? We will pick up beyond receptors and follow neural pathways up to the cortex, and we'll take a look at how the cortex represents your body. I will talk a bit about tactile perception and something called the 2-point threshold. I will mention J.J. Gibson's theory of active touch as opposed to passive touch, and we'll see the differences between those. Talk a little bit about temperature perception, a little bit about nobody's favorite, pain perception, and in particular, gate control theory, and tough to find a spot for this topic. It could go anywhere or everywhere. The topic is synesthesia. I'll explain what it is, and I'll talk about two current theoretical explanations of it, and I've also got a rather long video for you to watch of people talking about their experience of synesthesia. But first, somatosensation. Soma means body, so these are our bodily senses, which include the cutaneous senses. And the cutaneous senses are the ones that are found in the skin. And that includes touch and tactile perception, the perception of vibration, which, yes, does get separated out, texture, shape perception, warmth, cold. Yes, warmth and cold are separate. Itch and pain. Now, those might come as a bit of a surprise. Again, for many, many years, people just said, oh, our senses are seeing, hearing, smell, taste, and touch. Touch really doesn't cover it. We have a lot of other sensory abilities beyond just mere touch. Okay, so these are the cutaneous senses, and of course that's the focus of this lecture topic. I do want to mention some other somatosenses at the same time, though. And these are ones that are generally agreed upon. As I mentioned way back when, there isn't an official list of these are our sensory abilities. We can't get 100% agreement on this. There is some dispute. So these are the ones that are generally agreed upon. Proprioception means knowing where your body and limbs are with respect to each other. Kinesthesis is sensing and knowing the movement. So even with your eyes closed, you can bring your hand up and touch your nose, for example. You don't have to see it because you're relying on stretch receptors in your muscles. Kinesthesis, think about the word kinetic, and that should help you differentiate kinesthesis from proprioception, which is more kind of static. Haptic perception, interestingly, is sort of a higher order sense because it is a coordination and combination of tactile and kinesthetic sensation. So for example, if you're going to grasp an object, like for example, picking up an egg, so you have to sense when your fingers are actually touching it, and then you have to squeeze it, but not squeeze it too much. You don't want to pick up an egg and crush it and make a great big mess. So that is its own thing called haptic perception. And then I will throw this one in as well. I mentioned it in a previous lecture, our vestibular sense, or equilibrioception. Ooh, that's a mouthful. Our vestibular sense helps us to maintain balance. And I did have an image of the semicircular canals. This is a much better image. I think this, well, it shows it a little bit better. Again, they are kind of arranged at right angles to each other. So they help us to detect acceleration in any direction three-dimensionally. We also have a sense of just absolute spatial orientation, so which way is up, for example. So the vestibular sacs. So the vestibular sacs contain little crystals in there, and the movement of those is picked up by receptors within that, and then that helps us to determine which way is up, and the semicircular canals, again, for acceleration in any direction. There is something that I wanted to point out in the textbook. Now, in the glossary, the textbook does have an entry for the somatosenses, and in there, it includes the cutaneous system. It includes the proprioception and kinesthesis, but it does not explicitly say that the vestibular sense or haptic perception is part of the somatosensory system. There is no official list of what is or what isn't, but in looking through the literature, it's kind of generally agreed that the somatosenses do include the vestibular sense and haptic perception, so just be aware of that. So our cutaneous senses are housed in the skin. How much do you know about your skin? Well, here's some more. It is the heaviest organ in your body. It weighs about 4 kilograms on a person of approximately average size, which is 8% of your body mass. If you were to take that skin and kind of stretch it out and measure what size it is, please don't do that. You don't have to. It's been done with cadavers. Okay, it's just shy of 2 square meters of area. You're pretty sick if you think you want to stretch out the skin and measure. That's gross. Your skin is not uniform. It does vary in thickness. It is, of course, the thickest on the sole of your foot and the thinnest not just on your nose, but on the bridge of your nose. You know when you wear glasses where the glasses sit? Right there, that's the thinnest skin of your body. And that's where glasses are designed to sit. Great. Couldn't they design glasses to sit on the sole of your foot? Okay, never mind. Your skin could be glabrous, which means non-hairy, or hairy, which, is this a surprise, is also known as non-glabrous. See how that works? Okay, so non-hairy skin is found on the soles of your feet, on your lips, on your palms, for example. This skin, again, it's not uniform. It's not a plastic bag that holds your guts in, although that is one of its functions. It does have structure to it. It has layers. The outermost layer is the epidermis, and that can be broken down into the outermost layer called the corneum, or the stratum corneum. This is made up of basically dead skin cells. I mean, there's no way for nutrients to kind of reach that, so they kind of die off and they're kind of pushed to the outer layer. So if you're looking at skin, you're basically looking at dead tissue. I see dead people. Now your epidermis, being dead skin cells, those skin cells do slough off. Your epidermis renews itself about every 50 days or so. What happens to those cells that slough off? When you go around your house and you're dusting and you see all that dust, you know what that dust is? It's you. Okay, it's also bits of lint and other things, maybe dead bugs, but a lot of it is you. You shed about 90 pounds of skin in a lifetime. That's a lot of skin. Now, below the epidermis, which means epi means above, is the dermis. And why am I pointing all this out? Because it's meaningless trivia. No, it's not meaningless trivia. It's because at the boundary between the dermis and the epidermis, there are a lot of structures, which, notice how cagey I'm being? I say that they're believed to be receptors. And I'll explain eventually why I say believed to be. Okay, I have some diagrams. I prefer diagrams to photographs because the photographs are, honestly, they're kind of gross. So here's a chunk of skin. Hopefully you never see a chunk of your skin like this. If you do, you probably want to seek medical attention pretty quickly. So we have the epidermis, and then the dermis, and then a bunch of weird things in your skin. So they do differ in terms of their morphology or shape. We have raphini cylinders here, and here we have really big ones called Pacinian corpuscles, which can be just about seen by the naked eye, actually. There's a sweat gland, yuck. There's a Meissner corpuscle, and here we have Merkel discs or Merkel receptors. So lots of different weird structures. Okay, so let's start to go to an explanation of this. First attempted by specificity theory, which is based on the idea of specificity coding, first proposed by Max von Frey in 1894. Now, von Frey didn't have a lot of tools at his disposal. He could dissect things out and look at them under a microscope and try to make some guesses, which basically is what this is. Specificity theory proposes a one-to-one mapping for every different thing in the skin corresponding to a different basic sensation. So, according to von Frey, warmth is picked up by the Ruffini endings, cold by Krause end bulbs, which I didn't have a picture of those, touch by Meissner corpuscles, and pain detected by the nearly ubiquitous free nerve endings. Again, this is according to von Frey. I'm not saying this is the way things actually are. Well, we can experience more than just simply warmth, cold, touch, and pain. We can experience other kinds of complex sensations as well. Those, according to von Frey, are comprised of touch blends. And what von Frey did in kind of the great tradition of the 19th century, he stuck his hand into things and then kind of thought to himself, what does that feel like if I try to split it apart in my mind? So putting his hand into water, wetness feels like even pressure and cold. Whereas oily was weak pressure, warm, and then movement. It's very slippery, right? So wet and oily can be broken down into these kind of different components, and this is what makes them different. Furthermore, it was found that we have punctate, or literally point-like sensitivity. That is, we're not uniformly sensitive to, say, cold everywhere on our skin. It's not like that. We have certain spots that are sensitive to cold, but if you move a cold stimulus a few millimeters over, you might not feel it as cold at all. You might feel something touching you, but you may not actually feel it as cold. That spot might be sensitive to warmth or maybe to pressure or pain or something different. All right, so what we can potentially do to determine this relationship between the structures and our sensations is, well, it's a bit involved. We would have to first stimulate the skin, like you might get like a needle and warm it up and then touch it to the skin, not the pointy end, okay? You don't want to pierce the skin. And then ask the person, what do you experience? They might say, I feel some pressure, I feel something that's really warm, or they might say, I don't feel anything at all. Then what you'd have to do is right underneath that little spot of skin is figure out, well, what weird structure exists at the epidermal boundary. Okay, so it is theoretically possible. It does involve things like microneurography, which means you take an electrode, which is now kind of a pointy needle, and you stick that into an individual nerve fiber, and that can record when that nerve fiber is sending a signal, okay? Well, now we've got that, but what we need to do is a bit of reverse engineering. What is the weird encapsulated structure, the cell body, at the end of this axon? So what you'd have to do is, typically, using the same electrode, you now inject a tracer substance, which will be taken up, and it will go into the cell body. And then what you have to do is basically just take a knife and follow that axon, cutting through the skin until you get to that cell body. And then you can take a look at it and see what its shape is and what type it is. There are some problems with this. Would you want to sign up for an experiment like that? Yeah, we're going to be sticking a bunch of needles in you, and then we're going to be cutting up your skin, and we'll stitch you up again at the end, though, for free. And you'll get, like $5 an hour. Yeah, you won't find people who sign up for this. Yeah, I'd like to volunteer. No, not even if they're getting paid. It's pretty nasty stuff. So what is left for the researchers to do if nobody wants to sign? You can't do it with animals. They can't report the sensation verbally. So It's called self-dissection, but I think technically it should be vivisection. And if you don't know what the difference between those two are, well, you can go ahead and look that up. This basically means that the researcher would be doing this on themselves, cutting themselves open and shoving needles in and cutting the skin open, which that's not really pleasant. And you have to have a lot of motivation to do this in the first place. And that's not even the worst part of it. The worst part of it is the result of all of this showed no correlation between the structures and the sensations. You might find a cold spot, okay? Find the structure at the end of that axon, and it might be a Merkel disc one time and then a free nerve ending the next time. What about the next time? Well, you've only got 2 arms, and once you've cut your arms open, you can't really do it again and do a retest because you've cut the arms open, you've kind of destroyed the physical integrity of the skin, and even if you stitch it up and let it heal, it's probably a bit different than it was initially. So yeah, this is really not working out too well. Here's another problem. Let's say you get a stimulus and you touch it to the skin to deliver a very high temperature. The skin is not a static thing. It's very dynamic. It will absorb some of that heat energy, and it'll spread probably several millimeters, if not even centimeters away from the area of stimulation. Or try this. Just look at your arm and just poke your arm. Don't hurt yourself, but poke your arm and look at how much skin moves. You'll get skin that's many, many centimeters away moving when you poke yourself in the arm. You can't deliver, say, a touch stimulus to 1 square millimeter of tissue without it affecting anything else. It's impossible. So you're probably activating many dozens or hundreds or even thousands of receptors of many different types. So that's a problem. The cornea is interesting because it's innervated only by free nerve endings, no other weird little structures. But it has all 4 of Funfrei's basic sensations. Yeah, how does that work? And that's not even, I couldn't even fit it all onto one slide. We're continuing with the problems. The sensory spots changed over time. So if you do this research, you might find a touch, and you're not cutting open the skin, okay? You're just identifying the punctate spots. In the morning, 1 particular square millimeter of tissue might be sensitive to touch. And then in the evening, you try it again, and it's not sensitive to touch, but now it's sensitive to cold. What? What's happening? And lastly, one of my favorites called the thermal grill illusion, which I have very fond memories of doing it in elementary school. Kind of tough to do at home. What you need to have is 2 faucets in the same sink that are independent. And what you need is to have 2 hoses attached to each one of those faucets. One has really nice warm water, not really, really hot water, but warm water. And the other hose has really cold water flowing through it. You kind of twist them together, as it's shown in the picture, and then grab it. And then what you're going to do is let go really fast, because it'll seem to you like it's screaming hot, even though that's impossible. It can't be. It's simultaneously warm and cold. Now, this is not the same thing as taking some warm water and adding some cold water and mixing it together. No. That just kind of averages out. This is simultaneous warm and cold, which our brain interprets as really, really hot. How is that explained by specificity theory? It's not. So it's maybe not even possible to determine which sensation goes with which receptor. Not with absolute certainty because of all of these problems. What this shows us is that, okay, specificity theory, as intuitive and kind of simplistic it is not correct. So what happens is, well, the pendulum swings the other way, and the complete opposite is proposed, called pattern theory. This one says, even though we've got very, very different structures in our skin, they're all doing exactly the same thing. I know that's pretty counterintuitive, but the idea here is that each of these receptors fires at a particular rate. There's a certain number of impulses depending on the stimulus. So maybe if it's a very cold stimulus, maybe they fire at a very low rate. If it's like a lukewarm, maybe they fire at a greater rate. And if it's really hot, they're firing at their maximum response rate. And this is what indicates the nature of the stimulus, potentially. It is possible if we think about this. So if you think about mild pressure on your skin, you'll feel that as a touch. But as the intensity increases, as more and more kinetic energy is delivered to your skin, eventually that light touch is going to start to hurt. So it's touch that turns into pain. Maybe we can explain the coding of the stimulus in terms of the rate of neural firing, for example. But we are left with the question of why do we have all of these different kinds of receptors that look so different from each other? And they're all doing exactly the same thing? That seems pretty implausible. So maybe the answer isn't one or the other, so neither of these two extremes, but maybe the answer is somewhere in the middle. It's an idea called stimulus preference. It is an intermediate between those two extremes. So maybe it depends on certain receptors being activated by a particular stimulus and also their firing rate. So maybe the receptor structure is tuned to an aspect of the stimulus. It's a bit of a subtler notion. Let me explain. Here is actually some evidence that supports this idea of stimulus preference from 1966. Lowenstein and Skalak dissected out Pacinian corpuscles because they are the largest. It is relatively easy to see. You can almost see them with the naked eye, so 1 to 4 millimeters long, and they have a structure kind of like an onion, okay? So the actual nerve fiber is in the middle, and then it's surrounded by multiple layers of tissue here. There are those two pictures again. So why is there all this tissue surrounding that nerve fiber? Well, all of those different layers seem to absorb sustained pressure. Now, when pressure begins, all of that kinetic energy is transmitted to the nerve fiber in the middle, and it will send a signal when pressure begins. Now, as the pressure continues, well, that's absorbed by all of those layers, and the nerve fiber in the middle does not fire. But when you let go of the pressure, that will then cause the nerve fiber to fire again. There's a spring-back kind of effect. So the Pacinian corpuscle doesn't detect a sustained pressure, per se. What it does is it detects the onset and the offset of pressure. Or, if there's a changing stimulus, like a vibration, you can think of that as a series of touches one after another, and it will fire very strongly to a vibrating or changing stimulus. So it's not, again, detecting pressure, it's detecting an aspect of that particular stimulus. It is better tuned to detecting vibration. We do have evidence beyond just the Pacinian corpuscles that there is stimulus preference for those different structures that I showed you before. And again, it's kind of tough to study the structures themselves, so a lot of research effort does focus on the nerve fibers, which are a lot easier to study. So here is, for example, a pressure stimulus. It comes on here, it goes on for a while, and then it gets turned off. This is the response of an RA2 nerve fiber. That stands for rapidly adapting, and it is known that it connects to a Pacinian corpuscle, or PC. So it does have kind of some random firing, but it fires a burst at the onset and at the offset. And here's another separate nerve fiber. SA is for slowly adapting, and you can see it does fire throughout that entire duration of the pressure stimulus. So to capture this particular pressure stimulus, it does activate an RA2 fiber and also an SA1 fiber as well. So it shows this kind of relative specialization. It's not absolute specialization as proposed by von Frey. Okay, what we can do now is continue the path that the information is taking. It's been encoded. We've got some fibers now that are firing, and it gets pretty messy. So I am going to give you, I promise, a simplified version. So I'm going to give you a couple of major pathways, the spinothalamic pathway, and hopefully that makes a little bit of sense. So spino, meaning your spinal cord, and your thalamus. Okay. So this pathway is made up by the smaller diameter nerve fibers. Now they go up the spinal cord and your brainstem and eventually reach your ventral posterolateral nucleus, or VPN. And this is part of the thalamus. Sorry, I didn't make that absolutely clear. Okay, so there, I changed it. The VPN of the thalamus. However, it's a bit more complex. There are also connections that go to the limbic system, which of course handles emotion and memory. So what kind of information is being carried in this pathway? Temperature and pain information. Okay, that explains the emotion part. So if you're experiencing a painful stimulus, usually there is emotion that goes along with that. Temperature as well. So if you're too hot or too cold, that's really unpleasant. So again, we have an explanation for that because of this really direct pathway to the limbic system. Okay, the other major pathway is called the medial lemniscal pathway, and yes, this is actually self-explanatory. Okay, so we're using the larger diameter and longer nerve fibers that synapse in the medulla oblongata. They ascend via the medial lemniscus, and interestingly also go to the VPN, yes, of the thalamus. And this contains both touch and proprioceptive information. Okay, so of course the thalamus is not an end point. I don't mean to imply that, but we are now at the brain, and of course that means we need to take a look at the organization of the cortex, and specifically the somatosensory cortex. So the somatosensory cortex, the primary somatosensory cortex, is in the parietal lobe, and it is basically just behind the central sulcus, which is a deep groove. So both pathways do go to this part of the brain. Now here's what we know about this part of the brain and how we know it. So there's a couple of different techniques. We can use the evoked potential method, which means putting electrodes on a person's scalp, and then we can measure the electrical activity of what's going on in the brain on those specific locations. And what you can do is stimulate different parts of the person's skin. So you can tickle their feet or their hands or touch their shoulder and then look to see which electrodes are lighting up. This can work. There is a lot of background noise and it's pretty time consuming and not super accurate. If only we could get more accurate, pinpoint accuracy. It is possible. There are some downsides though. So this would mean directly stimulating the brain. So going in and directly stimulating tiny areas with very small jolts of electricity. This was done by Wilder Penfield, and he wrote about it in his book in 1950. Wilder Penfield was a neurosurgeon, okay, not a neuropsychologist. He was operating on people to remove brain tumors. And to do this, you have to be very careful. You don't want to cut out too much brain tissue because you might leave the person a vegetable or they might be paralyzed. So he really didn't want to damage any sensory or motor areas. But back when he was doing his work, this really wasn't known. What parts of the brain do what? So, okay, well, I'm there anyway. I'm in the brain. So basically what you have to do is under a local anesthetic, you have to basically take the top of the person's skull off and then deliver the jolts of voltage and then ask them what they experience because, again, it's under local anesthetic so that they can respond. So, yeah, they are actually awake. And they might say things like, "You touched me on my shoulder." Well, no, I didn't, but I just probed that part of the brain, so that part must be part of the somatosensory cortex that handles incoming information from your left shoulder, for example. So Penfield was able to map the brain, and one of the first to do that in a very systematic kind of a fashion. I have a video that I'd like to embed in here, although if there is a copyright claim on YouTube and it doesn't work, I'll have to link it separately.
Saul, toast is burning.
Toast is not. smell by probing the surface of the brain, we'll find the source of the seizures. Mrs. Gold, do you feel anything?
I can see the most wonderful lights.
And now what do you feel?
Did you pour cold water on my hand, Dr. Penfield?
Now what?
Oh,
What is it, Mrs. Gold?
burnt toast. Dr. Penfield, I can smell burnt toast. Dr. Wilder Penfield. He cured my seizures and hundreds more. They say he drew the roadmap of the human brain. We just called him the greatest Canadian alive.
Okay, so just to let you know, if you smell burnt toast, it doesn't mean that you've got a brain tumor. Okay, it was just this one famous patient of Penfield's. Okay, so the kind of big picture of what Penfield found was a particular kind of organization in the cortex. The connections aren't kind of haphazard. It's called a somatotopic organization. And that basically just means if there are two adjacent areas on the skin, so very close together, the connections from the receptors under those two similar adjacent locations are going to send their signals all the way up, eventually getting to the brain, eventually to the somatosensory cortex, activating neurons. And those neurons that are activated in the cortex are also going to be very close to each other. I do have a warning, I guess. My next few slides will be maybe NSFW, so they might not be appropriate. So if there are young children and you don't want them to see pictures of simulated nude human bodies, just be aware of that for the next couple of slides coming up. All right. So this is actually taken from Penfield's book. We have the motor cortex on the left and then the sensory cortex on the right. And they are on either sides of that central sulcus. So the motor cortex is in the frontal lobe. I'm going to skip that. I'm just going to be focusing on the sensory connections. Okay, so our backs are very large on our, actual bodies, but the amount of tissue devoted to incoming information from the back is very small. Your thumb, on the other hand, is pretty small, in the context of your body, but it gets a lot of brain tissue devoted to it. Look at your lips. Your lips are likewise very small. There's a lot of brain tissue devoted to incoming information from the lips. And then you can probably look and find the genitals for yourself. This is of a male. You can make your own conclusions about that. So the name that Penfield gave to this representation of how our brain feels our body is, there's a picture of it. The name that he gave this was homunculus. Homunculus, which literally means little man. Yeah, I know it's kind of a sexist gendered term. Sorry about that. So what someone has done is done a reverse engineering. This is the sensory homunculus. So constructing an entire three-dimensional model of a human being as felt by the brain. So for the brain, you're a lot of lips and you're a lot of hands and everything else not so much. Here's the motor version. So again, we have a lot of fine control over our lips and tongue and hands, so no surprise that our motor homunculus also has a lot of brain tissue devoted to those regions. And if you're ever at the Natural History Museum, they actually have some sculptures of the sensory and motor homunculi. Cool. I don't know if these look familiar to you. After seeing that homunculus, does it maybe look like Mick Jagger? No, is that just me? Okay, so to summarize, nudity is all gone now. So the general picture here is that some parts of the homunculus do not match the size of those actual parts on the human body. But the question that we need to ask then is why? Now, interestingly, the body areas that were really large on the homunculus, so like the lips and the fingertips, very large in the homunculus, not so big on our actual body. We're also very exquisitely sensitive there. Well, this could simply be to the high density of receptors that we have in our lips and in our fingertips. That might be part of it. But couldn't it also potentially be that we recruit more neurons in our cortex because we work a lot with our fingertips and our lips? Could that possibly be part of it too? And if that's the case, then this should be plastic. It should be changeable. A lot of studies looking at brain plasticity have actually looked at plasticity in response to changes in the body. So yes, it can be changed via experience. This is called experience-dependent neural plasticity. Some of the foundational research was done on monkeys. So what they do is they'd map the somatosensory cortex to the different fingers on a monkey paw. And then, I know this is kind of sad, but they would amputate one of the fingers. And then after a few days, they'd remap the brain again. And it turns out, well, the brain doesn't want to waste any brain cells. And if there's a part of the brain that was expecting signals to come from that now amputated digit, those cells would remap themselves and take over to responding to signals from other digits. And this would happen just in a matter of a few days. So the brain was highly, highly plastic. So I showed you a couple of homunculi. There was the touch homunculus and then the motor homunculus. We have multiple homunculi. There's a whole bunch of them. There's some for discriminating forms and shapes. There's another one for textures and so on, and maybe even some others that we haven't discovered yet. Touch, of course, is feeling a constant pressure, whereas vibration is a changing pressure. So maybe these exist along a continuum. So maybe touch is a single touch, and vibration, we can think of that as a series of touches. I'll come back to that. What we do experience is the kinetic energy. So if someone touches you with your finger, you might think, well, I'm feeling their finger. What you're actually picking up on is the kinetic energy that they're delivering to your receptors. Touch is important for development, social cooperation, and affiliation. You've heard the phrase touchy-feely? Yeah, touch is really important to have. It is really important for our development. And this has been demonstrated by Tiffany Field for many decades. In this study in 1986, they took premature babies and massaged them. The control group was where premature babies would typically be kept in an incubator. So it's a transparent plastic box. There's bedding in there, but there's a lot of lights and there's a lot of beeping and a lot of things, but you're not allowed to touch the preemie because you could infect them. So in this case, they actually got nurses all outfitted and gowned and sterilized, and they would massage these premature babies. What would that do? Just touching them in this kind of nice way. The outcomes were the massaged babies gained 47% more weight compared to the controls. They were more socially responsive and slept better. That is an important component. They were also discharged from hospital 6 days earlier at a saving of $10,000 per infant, and that was back in 1986 dollars. Now, when it comes to touch, we don't actually have to be touching, so I'm going to go beyond the physical contact and look at our personal space. So it's going into a little bit of maybe social psychology, but I'm allowed to do that. Edward Hall developed the zones of personal space, or proxemics, and it is the distance that you keep from other people depending on how you're interacting with them and maybe who they are as well. So I'll go through the different distances, starting with the most distant. We have public distance. This is used for public speaking. So when I'm lecturing, I'm usually at the front of the lecture hall, and I keep my distance from students, because if I were to stand right next to somebody and be in this professor speaking voice, it'd be really weird both for students and for me. And in an actual classroom at this point, I would actually demonstrate what that's like, and it's profoundly uncomfortable. Next, we have social distance. When you meet up with some acquaintances, you'll stay a certain distance away, so about 4 to 12 feet. Personal distance, this is for your good friends, this is for family members, so you'll let them get as close as about 1.5 feet to 4 feet away. If you're keeping more than 4 feet away, it seems kind of impersonal. Why are you standing so far away? And if you're closer than 1.5 feet, that's kind of wrong too, because anything closer than that is the intimate distance. And this is for whispering, for actually touching or for embracing your intimate partners. And again, we're most aware of this when there are violations of this. If you have to take public transportation and there are people in your intimate distance, that's kind of uncomfortable. Now, this is based on, I have to say, kind of a American Western-centric. This can vary among cultures. This isn't necessarily universal across all cultures. Most of the research has focused on the kind of Western cultures. It's kind of interesting that with the coronavirus and the suggestions that you keep 6 feet away from people, even 6 feet away potentially from friends and family members, it is pretty unusual, and some people have kind of reacted badly to that, at having to maintain such a distance from people that they're not used to being so far away from. And when it comes to touch, this next experiment, this is one of my favorites from 1984. The participants were actually waitresses at a couple of restaurants, and they actually enlisted them because they wanted to look at the effects of touch on tipping. So the waitresses were brought in and they were trained for different kinds of touch manipulations. There was a control condition in which the waitresses made very sure that they did not touch the customers at all. In another condition, they placed their hand on the diner's shoulder just for a specific length of time. It's not like it's there for 5 minutes, okay? And then in the C condition, touched the diner's palm when they were giving change. Okay, so this was in the early 80s when people would actually pay with like bills and coins, okay? So giving change was pretty common back in those days. I suppose you could do it now and handing back like a credit card slip or something. Okay, here are the results. The control group, the smallest tips of all the conditions. In group B, 18% greater tips. In group C, which is direct skin-to-skin contact, 37% greater tips. Now, there are lots of things to ask about. I did note that the participants were waitresses and not waiters. Who are they touching? Who are the people who are paying? Most likely they were men. And there is actually a body of research that does look at tipping. It's economics research, kind of beyond the scope of this course, but if you're interested, to give that a look. Here's some other findings when it comes to touch. Diners were more likely to take the suggestions made by a server when they were touched by the server. Again, like a touch on the shoulder, that kind of a thing. How about this one? Men who asked women for a date and touched them in an appropriate way, can I just say that? That's really important, were more likely to get acceptance. So twice as likely, but it's still only 20% compared to a 10% with controls. You want to get someone to dance with you? Again, these days, you have to be very careful about touching. Some of this research is from a few decades ago. Or how about this? If a teacher touches a student, more likely they will go to the blackboard to answer a question. Again, you have to be very careful with this. Or even, hey, can I get you to fill out this survey? It'll take you 10 minutes. Now, touch can be very nice. It can be sometimes very nice. Know what I mean? Know what I'm talking about? And I don't mean like on the LRT or something. So why is that? So a lot of research, and there's been a lot of talk about oxytocin in the last 10 years or so. This is what oxytocin actually is. It's a hormone synthesized by the hypothalamus and stored in your pituitary gland. And the reason why it has gotten so much focus is because it is released during contractions, during labor and lactation, but also released by touch and at orgasm in both males and females, and it may cause endorphin release. And again, there's a lot of research underway looking at some of its secondary effects. So what are these pictures of? Okay, so you can get scents, so colognes or perfumes that claim to have oxytocin in there, and you can attract someone or connect with someone. I don't know why they have a K in there. connect with someone. So I guess the idea is you spray this goop on you and you go up to people and they'll go, hey, I just have a connection with you. Yeah, I don't think it's quite that simple. So again, going to some of the foundational research from the 90s, they measured oxytocin levels in blood samples in 26 women, so a pretty small sample. Women got a relaxation massage. Where do I sign up for that? And they got paid for it too? Nice. And their oxytocin levels rose slightly, measurably, but not a huge amount, but still. They then also had another condition in which they asked them to remember a negative emotion or a negative experience. Interestingly, the oxytocin level fell, again, a little bit. How about remembering a positive emotion? And it is a recollection. So it's like go through it and feel the feelings and the smells and the sights and the sounds as if you're experiencing it again. So very kind of vivid. This one, interestingly, had no effect. So oxytocin response is pretty complex. Okay, let's go back to the cold hardness now. Get away from all this emotional touchy-feely stuff. Let's look at touch threshold. So there is a counterpart to absolute threshold. Remember that? Absolute threshold from way back when? Okay, the way that this is done is by using a von Frey hair. Notice it's von Frey hair, not von Frey's hairs. They didn't harvest them from von Frey. Okay. So von Frey was the first to develop this. I believe they are horse hairs, although I'm not completely sure. You have it embedded in this sort of stick, and what you do is you push this into the person's skin until the hair bends. And it actually bends with a very consistent amount of force. So what you could do is start with a really thick one and then kind of work your way to the kind of the thinner and thinner diameter ones until the person goes, I can't feel that anymore. Or the opposite direction, start with the thinner and work your way to the thicker and thicker ones. This is just absolute threshold. Okay, now once this has been established, what you'd want to do is take a look at difference threshold. But what would that be when it comes to touch? Well, one form of the difference threshold is a 2-point threshold, in which you have, 2 points that are touched on your skin, and you change the distance between the 2 points. Okay, so this uses a device called an anesthesiometer, and it looks kind of like a compass, okay? It's best to have the person either look away or have them blindfolded or something. And again, you get them closer and closer together until the person says, oh, I just feel that there's 1 point, and you can record that. This was measured back in 1968, and this is a famous diagram of that. They did males and females. It looks like this is the one for males. And they did the left side and the right side. You can see that left and right are not absolutely equal, interestingly. So again, a higher line means that people are less sensitive. Okay, so it's threshold is the inverse of sensitivity. So the lines that are lower means that people are more sensitive. So you're very sensitive on your fingers and your thumb, a lot less sensitive on your palm, as your upper lips, nose, and cheeks. Your back, however, is not terribly sensitive, and neither is your calf.